All right, this next video focuses on 6 antibiotics that target protein synthesis. So that is the process of translation, which is going to make all the proteins of the cell. Remember, proteins are the workers of the cell. Those are our enzymes, which we know are really important catalysts. And so antibiotics that target translation are lethal to the cell. So first up are the aminoglycosides, and what's going to happen here is the aminoglycoside is going to bind to this small ribosomal subunit, and which is going to cause issues when the ribosome tries to move down this piece of messenger RNA, right? So see how the ribosome is attached to the left-hand region of messenger RNA? Well, once it reads that messenger RNA and it adds those amino acids, it's going to scooch down to the right. That's called translocating. So the ribosome moves down the messenger RNA, reading those codons, building that polypeptide chain. So because the aminoglycosides bind to this small subunit, it messes with how the ribosome reads the messenger RNA as it moves, and it actually creates faulty proteins because there becomes this mismatch between the codons and the anticodons, and it gets a little chaotic. And so it results in faulty proteins, which results in cell death. Next up are tetracyclines, and these are going to prevent tRNAs from even binding to the ribosome. So tetracyclines also bind to the small ribosomal subunit, and you'll see part of the antibiotic kind of protrudes into that space where the tRNA is going to bind. So it prevents that tRNA from binding altogether. And that's important because if the tRNA cannot bind, it can't deliver the next amino acid that needs to be added to the polypeptide chain. So it essentially shuts down translation at that elongation stage. Next up are the oxazolidinones, and these are going to shut down translation from the very start because it prevents the initiation complex from forming. So remember, during initiation of translation, we need all 3 key players to get together. And so that happens first when the small ribosomal subunit binds to the messenger RNA. Okay, so that happens just fine. And then notice, though, that these oxazolidinones have attached to the large ribosomal subunit, again, messing up this complementary shape that's needed for the large subunit to come in and attach. And so it can't do that. And so because the ribosome can't even form, no translation is going to occur. Our 5th antibiotic is erythromycin, and this is going to bind to the large ribosomal subunit, and it's actually going to bind in such a way that the ribosome can't move. It cannot translocate down that piece of messenger RNA to read those codons and incorporate those amino acids. Okay, so again, this is going to be shutting down the elongation stage. Last but not least is chloramphenicol, and this is going to prevent peptide bond formation. So remember that tRNAs are bringing amino acids, which you see here by the AA at the top of the tRNAs. So the amino acids are attached at the top of these tRNAs, and normally a peptide bond forms between them. That's how the amino acids connect together. But notice where this chloramphenicol, this green rectangle, binds. It binds to the large ribosomal subunit in a very specific spot so that the 2 amino acids can't be bonded together. Okay, so again, it's shutting down elongation of translation. All right, we're going to go ahead and include the last antibiotic, which shuts down a different metabolic pathway, but it's the only one and it's the last one, so we're just going to attach it to this video. So folic acid is a really important precursor molecule to some important processes within the bacterial cell, like nucleic acid synthesis and protein synthesis. And so bacteria make their own folic acid. Animals, like us, do not. So sulfa drugs are great because they do not harm or even pose a threat to the eukaryotic host, right? And that is because animals do not make their own folic acid. We get it from our diet. So we don't even have the enzymes needed to make folic acid. okay? But bacteria do, and that's what we take advantage of with this antibiotic. And so sulfa drugs, their mechanism of action is competitive inhibition, and that's because it is a structural analog, or it looks very similar to an important precursor molecule called PABA. So if you look here on the left, there's PABA on the left. Okay, it's that ring structure with a little carboxyl group at the top and amino group at the bottom. And you'll notice in the middle, there's a synthetase, which is an enzyme. So PABA is the substrate. It binds to that active site of the enzyme, and it makes this molecule that you do not need to remember. But then it's going to go on and eventually make folic acid. Well, what the sulfa drug does, look at the structure of the sulfa drug and compare it to PABA. It's pretty similar, right? And so that's going to be competitive inhibition. And again, because the sulfa drug looks a lot like PABA, we call it a structural analog. And so it's just going to do normal competitive inhibition. It's going to bind that active site and block the actual substrate, which is PABA, from binding. So therefore, this folic acid synthesis pathway cannot be completed, and then the bacteria cannot survive and grow. All right, y'all, that is going to be the end of this video. Let me know if you have any questions.